fascinating areas in modern rheumatology, and it is bringing the different disciplines. Today, in this session, we will move from fundamental concepts and to emerging clinic applications in rheumatology and neurology. My name is Ertuğrul Çağrıbölek. I'm from Ankara. I'm a rheumatologist, and I'm glad to chairing this session with Professor McGonigal. And I'm giving the word to him to inviting the first speaker.
Thank you very much, Ertuğrul. So, yeah, lovely to introduce Mark. I know him as a great immunology academic, the only one from University of York. And then he moved to Oxford, where he's now one of 140 great immunology academics. And I'm hoping he comes back someday. So, Mark, over to you to talk about an interesting title here, How Evolution Shaped Human Immunity and Autoimmunity. And he's going to be addressing this not from a Darwinian perspective, but from a rheumatological, anatomical perspective, I think.
That's correct. All right, thanks. So I want to tell you a little bit about the embryological origins of site-specific arthritis. It's a shout-out for the postdoc who not only did the science, but actually went to art school before she became a scientist. And you can see his lovely drawing here. And it's really focused on why the joint specificity. So a lot of you know about inflammation location. This is very pathognomonic in rheumatoid arthritis, that it affects particular joints than others. Yet the disease itself in humans is actually a sort of a form of a vasculitis, i.e. antibody-antigen complexes end up driving the initiation of the arthritis. Yet it affects particular organs, particularly with comorbidities, but also specific joints. So you have a very typical pattern of symmetrical PIP joints. In deference, you get distal sparing, and rather psoriatic arthritis is the other way around. You see the distal joints, but you less commonly see PIP and MCP joints. And so the question is, what is the origin of this? Because if it's a circulating autoantibody-driven pathology, why then do you see these joint specificities? And I want to go on to the role of anatomy in site-specific arthritis. And the reason why we really need to understand what a normal human joint looks like, and actually OA is not a normal joint, I'm afraid, that lots of studies use OA as controls, is that we need to be able to reset immunity and we need to cure immunity. In order to do that, what we need to do is not just drive things down to a clinical remission, we need to actually develop a next-generation therapeutics which lead to a molecular remission of the disease, not just a subclinical disease. And so we need to get insights from development and from the adult joint in terms of how you return to health and how you get drug-free remission through joint repair. And one of the ways to do that is really understanding the concept of distant to normality. In today's talk, I'm going to focus on the normal joint development of function. The way we do this is using a single-cell approach, looking at the 200 joints in the humans, across tissues, across diseases, when we have atlases of over 50 million cells. We obviously focus on diseases of interest to us, RA, PSA, JIA, inflammatory bowel disease, and other rheumatologically related diseases. And what we do is we look at the proportion of different cell types in the healthy, and then look at what's going on in different forms of inflammatory disease, and understanding what would it take to get back to the healthy state using single-cell genomic approaches. And I'm going to use that today in development. And one of the key things is it's a stoichiometry of the different cell types, which is really important to understanding health and disease. And that's also that stoichiometry of different cell types is very important on the susceptibility of different joints to getting different arthritic pathologies. And so the way we're going to do this, because we work on humans, and I'll go through why we work on humans in a minute, is that we use spatial transcriptomics, we use single-cell RNA-seq, more classical 10x, we use a multiplex proteomic, and the great part of that is we can also use ancestral material, and we do a lot of 3D anatomy work, and one of the, actually a summer student was playing around with one of the 3D rendering systems that we use in the lab. And so one of the reasons to study human fingers is that mice don't have fingers. So surprisingly how many reviewers try to tell me about how toes in mice are the same as human fingers, which they are not. I also like to remind them that the last ancestor of a mouse and a human was around when T-Rex was eating our ancestors. And so that's a problem because actually 100 million years is a lot of evolution, and mice were built to live under your refrigerator and to replicate and be eaten by cats and snakes. They were not driven by human pressures. And humans have something that's very unique to us that we all know, which is fine motor control. We are able to use tools with power and precision. Even our great ape ancestors do not have precision grip. They could pick up a stick, but they can't use it as a precise tool. They couldn't write. That's not a property of their brains. It's a property of actually their anatomy. They've developed for a very different purpose, and a purpose that fits them very well. And only humans spontaneously get rheumatoid arthritis. Our great apes have all sorts of other rheumatological diseases, but they do not get rheumatoid arthritis. And so the only mouse used in this presentation was my computer mouse to create these slides. And that's actually very important to remember. I additionally always tell people, because I do quite a lot of quantitative system pharmacology in the image space, is that we need to have rational drug development, and I call it falling into the mousetrap. And that's particularly common. It's not that we don't find models of rheumatoid-like arthritis in mice useful, but they often work by a completely different mechanism than is driving the human's disease. And I often say mice lie, monkeys exaggerate, and humans always tell the truth. And part of this is the biology here is not conserved. And we also need to remember that humans are a masterpiece of design. Our hands are immensely good precision and power grip, able to throw spears and do all sorts of things humans can do. But actually, we really need to understand human anatomy, and there are flaws to that anatomy, or trade-offs, which are evolutionary, and some are just a product of evolution. And one of that is the differences between our DIP and PIP joint that allow us to do clawing in our DIP joint and precision grip in our PIP joints. And so the way we've gone about this is through understanding development of the human joints. And what we've done is taken the DIP and PIP joints from week 8 through week 16 in human development. We've isolated the joints. We then enzymatically digested them, used 10x single-cell RNA-seq. And you can see all sorts of population. It is mostly a stromal fibroblast material at that stage, and there is some differences, but really until you dive into the biology, there isn't much difference in terms of the cell proportions in DIP versus PIP joints. And using the single-cell RNA-seq, we're able to identify all the different populations and different fibroblast subsets, and we can use a series of markers to delineate chondrocytes away from other fibroblast lineages. We can look at different progenitor populations. For instance, CLU stains a particular population of lining layer and chondrocytes, and TTP3 is a really good marker. And we're able to use confocal microscopy and RNA scope just to verify that these populations are unique and they have unique pieces of anatomy. And we've learned a lot about the biology from that. And one of the nice things, of course, at this stage is that the joints haven't been in much use. Consequently, actually, there's a really nice alignment. It's not messy, as you would see in an adult, because you're always damaging your joints, between location and gene transcription. And indeed, these days we've gone on and done this using spatial transcriptomics. We mostly use Xenium because it's a very forgiving technology, and then we pile on another 100 antibodies with phenocyclofusion. It really allows us to identify all the populations from the skin through the synovium and understand the architecture of the developing human joint. And we found this particularly powerful in understanding what is going on. And indeed, one of the things we were able to identify is what drives lining layer fibroblasts, and that's driven via a gradient of EGFs, HB, heparin-binding EGF, and also hypoxia. And what we're able to show is transcriptionally, you can see the effect of various transcription factors in a lining layer, soft tissue fibroblasts versus the cartilage. And particularly, you get, as you go through the different populations, you see this hypoxia occurring towards the lining layer. And indeed, when you use a hypoxia probe, look at versus the endothelial cells in purple, the CD31 positive endothelial cells are quite distal from where you're seeing the hypoxia in the interzone and in the developing synovium, and that is validated by, you also see the expression of EBGF in the developing synovium. And if you take the embryonic synovial fibroblasts, you culture them, and you give them HBGF, and you give them BTC, also works in vitro, and you put them to hypoxic conditions. H is for hypoxia. You're able to induce the PGR4, which is a lubricin in the lining layer fibroblasts. You're also able to upregulate the EBGF. So there's a loop of EGF receptor signaling and hypoxia, which is driving the lining layer fibroblasts. The other population in the lining layer is actually a very fascinating population of fibroblasts called PI16 fibroblasts, and they are highly enriched in the PIV versus the DIP joint, and that's shown there in this F2 population. And there's a big difference between the amount of these PI16 positive pro-inflammatory fibroblasts, which are later involved in human disease later on, and between the PI and the DIP joint. And what we've done is multiplex imaging and looking at the PI16 fibroblasts and looking at the topological correlation between PI16 expression and the different parts of the synovium. And what we're able to show is that the PI16 fibroblasts are really located around two structures. One of the blood vessels, and we'll talk about vasculature in a few minutes. So where you see lots of vasculature in the sublining synovium, developing sublining synovium, but not in the lining layer synovium. And you also see it around the tendon capsule borders, and I'm sure Dennis will tell you that tendons and enthesis are very important in arthritis, and thus we find these pro-inflammatory fibroblasts from the embryo onwards, which are associated with those structures. And what we see, again, is the proximal joint. There's a statistical increase in these PI16 positive fibroblasts. So are they functionally distinct? So what we did was take the DIP versus PFI joints. We can't use PI16 because it's an enzyme, an ectopic enzyme, to sort cells, but what we can do is use a proximal marker, which so happens to be CD34. CD34 positive, Thi1 positive fibroblasts are PI16 positive, Thi1 positive, CD34 negatives are PI16 negative. We sort them and we stimulate them with TNF and IL-1, and then we look at the gene transcription. And what we see is there's a very distinct difference between PI16 positive fibroblasts in vitro and PI16 negative fibroblasts. And these PI16 positive fibroblasts, they respond by making immunological molecules which are associated with immune regulation, so they become inflammatory, and vice versa, and in their resting state, they express lots more adhesion molecules. So they're quite a different population of cells. And again, you can look at a large number of processes from immune responses, endothelial migration, etc. So all of these things are regulated and they respond very differently to the fibroblasts that you see in the lining layer when hit with inflammatory signals. So we believe, and I've said earlier, that there's a big difference between the PIP versus the DIP joint. And so we wanted to look at this in 3D. To this, we went to the Diamond Synchrotron and worked with Peter Lee, Sharif Ahmed, and his colleagues at Diamond Synchrotron, looking at the three-dimensional structure of this. And we're able to, of course, use the synchrotron energy to image through the sample in 3D. We then want to see, is there a difference between the PIP and DIP joint? And what we really notice is this big difference in the 3D volume of the palmar side of the PIP versus the DIP joint. You don't see this lot, and that's your inside your joint. And that's probably very important in that control of precision grip. And it's highly vascularized, and we'll show that in a second. So the palmar synovial volume is very different, and it has a slightly different density between this additional palmar synovium you see in the PIP, which doesn't exist in the DIP joint. So what we find is that the dorsal side, there really isn't any difference between the synovium between the DIP and PIP, but the big difference is in the palmar side of the synovium. And again, this relates to PI16 and VWF staining. And so we wanted to take a better look at what's going on with the vasculature. We need to develop a better approach to looking at vasculature, so I'm going to show that here. So what we do is we take the 3D joint synchrotron imaging, we create image stack, and what we've done is trained AI on these sections, and there's a level of annotation that occurs there. And then this is run through a machine learning model, and what it allows us to do is create an XY inference stack of different cell types, and actually we can do additional information, which I'm not going to talk about today, when you bring different forms of imaging together. But you can create three-dimensional volumetric rendering of feature extraction using these automated AI-based platforms. And to do that, we've looked at the vasculature in PIP versus DIP joints. And what we can show is there's quite a distinct difference between the vascularization associated with the synovium in the PIP versus the DIP joint. And that additional vasculature, if I take you back to the beginning of the talk, is potentially associated with these antigen-antibody complexes stimulating these PI16 positive fibroblasts in the adult joint to drive a rheumatoid arthritis phenotype. But they're there from the beginning. You're anatomically predisposed to getting rheumatoid arthritis in the PIP joints because you have this ability to have that precision grip. At least that's the hypothesis we like to put forward. And indeed, Sarah very beautifully looked at the relationship between the fetal cells and the adult fibroblast populations. And again, we can get a nice map between the different populations in the embryo and in the adult. And thus, actually, a lot of the information we're using now is really useful in terms of understanding the development process required to repair particularly the lining layer, but also the return to normal in terms of the sublining layer. And it really comes down to the stoichiometry of the different cell types. So this really comes back down to this concept of inflammation location. There's a cellular molecular basis for this. It's based on human anatomy. There's a key role, difference between the anatomy of the DIP and the PIP synovium. We have a key role for the PI16 cells involved in disease tropism and progression. There are anatomical differences between the PIP, and thus that probably predisposes humans to be at risk for RA. When we have done some primate studies on the fingers, we haven't seen this difference, although that's been very early stages. So we have this hypothesis that neurovascular structures are required for the evolution of power and precision grip, but there's an evolutionary trade-off which our ancestors probably didn't spend a lot of time worrying about, which is rheumatoid arthritis. I just want to thank the team, particularly Sarah, I see hiding over there, and Max, and the entire team, because these take a massive team effort. The groups at UCL, particularly Peter Lee's done amazing work on 3D anatomy of joints, along with the Diamond team, and the whole group in the Kennedy. And if anybody's interested, of course, of visiting the Kennedy Institute, particularly myself, I share a group with Chris Buckley and Callie Dendrew. So lots of fun there, and I'll stop now. Thank you.
Thank you, Dr. Coles. We can open the questions. Any questions for Dr. Coles?
Thanks for excellent presentation. My quick question is, how did you manage to get the cells from the embryonic DIP, PIP joints of the humans?
Yes, so the UK is particularly privileged to having a human developmental biology resource. And we work with UCL and receive material from UCL. And so we're able to get early stage consented embryos to study the developing finger joints.
So they may perhaps aborted fetuses?
Yes, that's correct. Yeah.
Thank you.
I may kindly remind that you can also EULAR app to ask your questions from the Q&A part.
Yeah. Mark, it's really surprising that this is there so early in development, the difference between the DIP and PIP. Because you mentioned the DIP in adult, we use it for grasping and power.
Yep.
The PIP is more finesse, but in an embryo in vivo, in a a water-surrounded safe environment,
so
you wouldn't expect those differences to be already manifesting.
So that's interesting that humans, that's very much true. You spend most of your time in the womb developing your brain, not your immune system or your joints. So we're very brainy creatures, but actually the differences are there from the very earliest stages of joint development, and there's probably involved various mutations in the development of hominids, which led to these changes and so it's embryologically driven, these differences, because we need to have a particular anatomy to have a particular function.
So you're differentiating there between psoriatic and rheumatoid, which is very interesting. In regards to the monogenic autoinflammatory diseases, paradigmatic examples, FMF and TRAPS, the arthropathy is more a large joint predisposition and not small joint. And FMF in particular is neutrophils. And there's obviously, you didn't talk much about the immune development of the joint. You focused more on the vascular and fibroblasts. So is there any difference in the immune composition, like macrophages?
So there's relatively little, there is some macrophages there, and that's important. And Stefano actually showed a little bit of data yesterday on the development of the macrophages in the lining layer in the developing human embryos. But these different JIA and some of these rare mutation-driven monogenic diseases are very different in terms of the biology driving them. There's a very beautiful paper by Chrissy Bolton, working with Lucy Wedderburn and Adam Croft and myself, looking at JIA and mechanisms of that. That's published in Science Translational Medicine. But psoriatic arthritis have a very different immunological mechanism behind it. It is not an antigen-antibody-driven disease pathology. So these very different, they can appear similar, but actually they're completely different immunological diseases.
Yeah, no, it's fascinating. So things like citrullination in the tissues, and then immune complexes and autoantibodies, and then inflammation. You're basically saying the DIP seems to be protected from the earliest stages.
Relatively. We've discussed that you've had a couple of patients with DIP rheumatoid, but in relative terms it's rare. You still see Pi16 positive fibroblasts and the vasculature in the DIP joint, but it's quite different from the PIP joint.
Apologies, I'm blinded by the light. There's two questions, sorry.
No problem. Thank you for the fascinating talk. Enrico Fuzzi from Stockholm. My question is entirely out of curiosity and ignorance, likely. So, thinking in terms of like natural experiments, I'm thinking of humans with no PIP joints, like proximal symphalangism or stuff like that. Do you know if they ever get RA, for example? Are you aware of any literature on it?
I'm not aware, so yeah.
Thank you. I always wondered about the difference between DIP and PIP joints. So did you look at toes as well?
So we haven't looked at toes. We have a famous saying from my collaboration with Chris when I used to share the office, that the reason why rheumatologists don't look at toes is because they smell. But in all seriousness, we decided to focus on the finger joint first, but I think that toe joints are interesting, and they do get rheumatoid. So, it's not that we're trying to avoid them, but the fingers was kind of a very clear-cut story. So, yeah, that's the only reason we haven't looked at those yet.
Yeah, so that's fascinating, this demonstration of how autoimmune disease topography and psoriatic, which is more auto-inflammatory, are really there at a very early stage. So, yeah, thank you so much. In the interest of time, we'll keep the show on the road. Ertugrul and myself were talking about introducing speakers, but since I work with Sanisha in Leeds, I felt it would be easier for me to introduce him. He's an immunologist, and he has a big interest in autoinflammation and immunodeficiency, and he's going to present work on somatic mutations beyond vexus, where he was one of the original vexusologists. Thank you, Sanisha.
Thank you, Dennis. Thanks for invitation. It's a bit of a disparate session. This we have a bit of evolution, somatic mutation, autoinflammation, then AI. So I'm squeezed in the middle to talk about the autoinflammation and somatic mutations, which is just appropriate. So I thought, first of all, I mean, it's a mixed audience. I presume most people are familiar with the concept of autoinflammation. I guess this is why you're here. But I thought just to remind us all what we mean by autoinflammation, because it's relevant for the rest of the talk. And it's the group of diseases which is now coming into its own. It's about 25 years since it's been originally defined as an autoinflammation as a concept. And it's these diseases which are caused by the dysregulation of the innate immune system. And it's really the myeloid cells from the innate immune system that are the key effector cells and key players in these diseases. And its production of these pro-inflammatory cytokines from the innate immune system together with the role of the myeloid cells that causes the disease and causes the symptoms and pathology. So these patients will present with a systemic sterile inflammation, which can be either periodic, and some of these diseases do have this very periodic features, and that's why they're called periodic fevers, but quite often there is a continuous inflammation and continuous damage. So most of these diseases for which we now have genetic basis do tend to present in early childhood. And these days, our pediatric rheumatology colleagues are excellent in recognizing these diseases. So it's very rare for us in adult medicine to actually find any patients who haven't been diagnosed already with one form of autoinflammatory disease. And there's now over 50 separate genetic causes of autoinflammation. But that is not to say there's not plenty of autoinflammation in adult practice. And there are plenty of patients who are presenting with autoinflammatory-like clinical picture, and now conditions that we are beginning to define in adult medicine that do have the same pathogenesis. So thinking, usually when we think about genetically determined disease, actually we talk about inborn errors of immunity where the autoinflammatory diseases are part of the whole family. Often we think about patients who have inherited mutations either paternally or maternally, or if they have homozygous mutations from both parents. Quite often these mutations are de novo at the beginning of the organ development, but actually these mutations can happen at any stage of zygote development. And actually what we're now learning that these mutations can now happen quite often even in adulthood and beyond kind of early childhood. And the immune system is very well placed to be particularly targeted by somatic mutations because it originates from the bone marrow. Bone marrow is the organ which continuously replicates. Most of the immune cells still are produced from the common hematopoietic stem cell progenitors. It's renewed from this source within the bone marrow. And clearly, it's not surprising that you can get mutations occurring at later stages of development at different level of the bone marrow evolution and different immune cells. And for us, in autoinflammatory field, of particular interest are the mutations which may affect the monocytes and neutrophils and so on, which are the effector cells that are going to be mediating some of these conditions. What this does occasionally is that we can get diseases that are actually complete phenocopies of the inborn errors of immunity. So you can get the disease which is due to somatic mutation somewhere alongside of the immune evolution and actually look exactly the same but presenting later in life. Or these changes that can happen later in life can affect the way how the other common diseases may present. And one of the first examples of this was this NLRP3 late onset autoinflammatory disease. And this was well described. The gene was discovered in 2001 by Hal Hoffman, that it's the NLRP3 inflammasome which causes this whole group of autoinflammatory disorders called now NLRP3 autoinflammatory disease spectrum. And here, due to gain-of-function mutations in the NLRP3 inflammasome, you get overproduction of IL-1 beta, which in turn causes all these other symptoms which are very recognizable. And the disease severity, it's in a way related to how much IL-1 beta do you produce. But then we and others have found these patients in clinic turning up in their 40s and 50s and 60s with exactly the same disease. So basically, no prior history. Most of the inborn errors of immunity and CAPS you present usually in early childhood, and we had people presenting with exactly the same phenotype in their 50s. And what turns out to be that these patients have acquired mutations within the bone marrow affecting the same inflammasome, and they were gain-of-function mutations, and the range of mutated cells didn't need to be huge. Between 2 and 10% of myeloid cells within the bone marrow was sufficient to cause the disease. And the whole concept was also replicated in murine studies where neutrophil-targeted mutations were sufficient to replicate the full phenotype of the disease. And why should this happen? Well, it's surprising it's not happening more often. And this is an interesting, nice work from Sanger Institute, which shown really that the rate of somatic mutation starts from childhood. As soon as you're born, as the HSCs are replicating, the mutations are there, they're accumulating. It's surprising, really, that we survive all that well throughout the adulthood without much else going on. And some of these mutations are quite damaging. Interestingly, most of these mutations will affect the HSCs rather than necessarily any other progenitors down the line. So in essence, as you get these mutations, more likely to affect various lineages. Most of the time in the bone marrow, and this is now the language we all needing to learn and expand beyond our all ologies and specialties and so on, is because the medicine is becoming more complex and there's more overlap between what we do, is that these mutations cause the so-called clonal hematopoiesis of indeterminate potential and hematological malignancies. So within the bone marrow, as these mutations accumulate, what they do, they give survival advantage to some of the progenitor cells. And these progenitor cells, if they acquire further mutations, will potentially then go on to develop hematological malignancy. So there is all these concepts that we all had to now become familiar with, even though we're not necessarily working in hematology, but these are the changes that are happening to patients in their later years and which are relevant to inflammatory diseases, as I'll come to show you. So the clonal hematopoiesis of indeterminate potential is simply the state where you have some of these mutations which haven't really caused obvious disease yet, but they've caused some expansion of the clones. So the bone marrow is already becoming clonal. And then you have the next stage where you have maybe small degree of anemia, which is called CCAS, and then further progression into more kind of hematological malignancy state. Eventually, you may progress to acute myeloid leukemia. But what's quite interesting is that, and again, this has been discovered due to large sequencing studies and epidemiological work, that this CHIP state is not uncommon. It's quite common in patients over age, individuals, not patients, over age of 70, where about 10% of individuals will have some degree of CHIP. But it's not entirely benign situation as we used to think of it, because obviously it hasn't caused anemia yet, but it's been associated, again, epidemiologically, with a whole host of chronic diseases and chronic inflammatory conditions. So, coming back to rheumatology, why is all this different, important to us and in autoinflammation? Well, we noticed, and together with Dennis in clinic, we had a few of these patients who have turned up, again, in their later years with a picture which really looked like autoinflammation, but we just couldn't find any of the autoinflammatory genetic mutations to explain that. But quite often what was observed that many of these patients were under hematologist for some sort of mild dyscrasia, maybe it's just degree of anemia and so on. And we were interested to then, given obviously that in myelodysplastic syndrome and other related conditions, it's the myeloid cells which are the key cells that are affected, we wondered whether or not that has a connection with some of these autoinflammatory manifestations. And this is retrospective work which we published a long time ago. We just show that, in essence, the autoinflammatory-like clinical picture is far more common in patients who have mild dysplastic syndrome, and actually it's associated potentially with worse prognosis. But the condition which really brought this home, and really, the concept of changes within the bone marrow and autoinflammation and many of these inflammatory disorders, which really finally cemented this concept fully and allow us to really pursue this further, is the VEXAS syndrome. And VEXAS syndrome was discovered in 2020, and it's a condition which basically is due to somatic mutations that affect myeloid cells predominantly. It stands for vacuoles because this is a typical appearance of the mutated cells within the bone marrow. E1 enzyme, which is the function of the gene that's affected in VEXAS syndrome, X-linked, is because the gene, the UBA1 gene, is on X chromosome. A for autoinflammatory because patients present with autoinflammatory symptoms, and S for somatic because it's an acquired phenomenon. And this is a condition which largely affects elderly men because the gene is on X chromosome. And I don't have necessarily time to delve too much into the pathogenesis of VEXAS, but in essence, what happens here, again, it all starts in the bone marrow. You get a mutation at the HSC level, so hematopoietic stem cell progenitor at the top, and the mutation is quite devastating, actually. These mutations are not, they haven't been described in childhood because they're incompatible with life. But in this situation, because they're occurring on a somatic level and only affecting some cells, there seems to be some potentially survival advantage to these cells within the bone marrow. And what seems to happen is the cells that carry the mutation create this niche of inflammatory niche within the bone marrow, which seems to protect these cells, but actually the wild-type progenitors are then subsequently replaced over time. Interestingly, when you look down the stream in terms of development, it is only really the myeloid cells, the myeloid lineage, neutrophils and monocytes that can exist with these mutations in periphery. So all the immune cells, once they leave the bone marrow, the lymphocytes, B cells and T cells tend to die within the bone marrow if they carry the mutation, but the cells that are present in periphery are neutrophils and monocytes, which then cause most of the damage. And this is illustrated here, really, which shows the electron microscopy picture of the VEXAS neutrophil, which is clearly disordered and abnormal. And if you study this cell in detail, you find all sorts of inflammatory pathways are activated. And actually, a lot of the manifestations that we see in VEXAS syndrome in terms of inflammation are due to these mutated cells, either neutrophils or monocytes, infiltrating the target organs. And this is what gives you the picture of the VEXAS man, which is basically, VEXAS is really now taken over from lupus and other conditions as a true mimic of many conditions, and it goes beyond rheumatology, and these patients can present to dermatology, hematology, and all other ologies, and this is why we all have to start learning different languages to understand and how best to manage some of these cases. So most of the common manifestations are these skin dermatological manifestations due to, again, mutated cells infiltrating the target organ. And also important to notice that there is a significant hematological component. So some of these patients do develop frank mild dysplastic syndromes and other hematological diseases. So this is why they need to be really managed jointly across specialties. But what's the story now beyond VEXAS? VEXAS has really given us this idea that these things do happen, and we now increasingly seeing more of this in clinic. Clearly, it's not a rare condition. We estimated in the UK from the work that we've done, there's about 172 new cases of VEXAS syndrome in England in population of men over 50 who have been investigated for hematological malignancies. And we have now certainly identified multiple cases across many centers in the UK and beyond. So we continue to see these kind of patients in the clinic, and these are two cases that we had been looking into recently. which kind of started our journey again beyond VEXAS, looking at other somatic mutations. And these two patients presented again with a very VEXAS-like picture. They had this disparate inflammatory condition where they had some fevers, weight loss. One of them developed polychondritis, the other one developed more like a polymyalgia rheumatica type picture. They both had very little of anything in terms of hematological manifestations at that time, and they're both men in their 50s. And what we found in these two patients, they shared two mutations which are known to the world of hematology. So this came through, because we were looking for VEXAS, we did a myeloid panel, and we found two mutations in two genes. One is the IDH1 and SRSF2. The other patient had IDH2 mutation and SRSF2. And these mutations are known to be pathogenic already and being described before. And the first step was to then ask a question, is this behaving similar to VEXAS in the sense that these mutations are largely confined to myeloid compartment, because that would give us some idea that this is probably maybe following similar pattern. And indeed, when you do the cell sort and you look for, check for where the mutations are, they're largely found in the myeloid department. And then, we are interested how both of these mutations can contribute to the inflammation, but we focused largely on IDH1 and IDH2 because there was a little bit more known about this, and actually there are potentially some targeted therapies for these mutations. And these mutations are known beyond hematology as well. They also cause neurological tumors and so on. Interestingly, so IDH, and again, you have to go back to your Krebs cycle and basic biochemistry. These mutations, these enzymes are found the part of the Krebs cycle which allows obviously glucose and other compounds to be broken down and ADP to be generated and ultimately energy generation. And the IDH2 is found within the mitochondria, IDH1 is found within the cytoplasm. They essentially do the same thing, which is the catalyzed conversion of isocitrate to alpha-ketoglutarate. But if you have a combination of the wild type, and these enzymes are multi-meric entities, so if you have a combination of the wild type and mutated enzyme, you have this other enzyme which doesn't convert into alpha-ketoglutarate, but into 2-hydroxyglutarate. So a different kind of metabolite entirely. And this is called oncometabolite, and it's been shown to maybe have some role in propagating malignancy, but we didn't really know what the role of this was really in inflammation. And next, what I'm going to show is really work from Sam Reiner, who is in my lab postdoc, who has been working on this very hard. We are close to hopefully getting the story completed. But just before I show you the data, just to remind you why this is important, and this immunometabolism story, which is now beginning to kind of take more central role in some of these inflammatory situations. So this is work, Luke O'Neill and his team has shown a long time ago now that when you stimulate M1 macrophages with LPS or interferon gamma, you get a switch in terms of how the Krebs cycle operates. And you get two exits. TCA cycle is broken. You get accumulation of citrate, which then allows you to produce this other compound called itaconate, which is anti-inflammatory. On the other hand, you have also accumulation of succinate, which then allows you to then stimulate IL-1 beta production through activation potentially of NLRP3 inflammasome. So we looked at these metabolites, well, not me, when I say me, some looked at the metabolites after he had stimulated the cells from the patients with VEXAS syndrome and healthy control and the patients with these IDH mutations. And what you can see on this hand here, that the IDH mutations, patients with IDH mutations do not make any citrate. It's kind of, from that point of view, healthy controls and VEXAS patients do make citrate as you would expect, but these don't. And actually what that leads to is also reduced levels of itaconate, which is the anti-inflammatory metabolite, which you would hope would be there to provide some negative feedback. On the other hand, these IDH patients make much more succinate than the VEXAS patients have healthy control, and again might provide much more supply of this intermediate metabolite than to drive the inflammatory activation of inflammasome and IL-1 beta. In addition, obviously, these IDH patients make this oncometabolite, 2-hydroxyglutarate, and we were wondering, does that actually do anything on its own rather than just being an oncometabolite? And this is an experiment, again, done by Sam, where he added oncometabolite to healthy control monocytes, which he differentiated into two types of macrophages. And in essence, what it shows is that oncometabolite, together with LPS, potentiates your normal response to LPS and so on. So you produce much more of IL-1A, TNF, IL-1 beta, and IL-1 beta production is also dependent on the NLRP3 inflammasome. So altogether, it shows that, you know, for reasons which we're still working to try and fully break this down in terms of why it causes the inflammation, but there is certainly significant evidence here to suggest this oncometabolite is important in driving of some of these inflammatory phenotypes. And to finish off and just give shout out to Leon, who is also in the audience, the other postdoc in the lab who has been working hard in terms of trying to, you know, bring the role and world of somatic mutations beyond autoinflammation. We were interested, obviously, to know whether or not this phenomenon is applicable to other rheumatological diseases and actually wider inflammatory conditions. And we are lucky in Leeds that we have got these really amazing cohorts that many of our colleagues have spent years and a huge amount of resource to collect. So we have these patients who don't, you know, pre-RA, so they haven't got rheumatoid arthritis, but they're CCP positive. And they've been followed to up to 10 years to see who is going to progress and who is not going to progress. And this is only part of the multi-omic study that we have done. But what this shows is that basically the patients who have gone on to progress from pre-RA have actually got enrichment of these clonal hematopoiesis mutations comparing to the non-progressors. So this phenomenon concept is not only really reserved for the autoinflammatory disorders, but probably has wider implications. And I'll leave it there with some just concluding thoughts. You know, I think the somatic mutations are beginning to take a bit more of a central role in terms of how we think about the immune responses, and particularly in aging. We are all getting older. You know, I'm getting to the age now where I could get Vexer syndrome very easily, so it's important to, you know more about this. And, you know, I'm sure we're going to find more of these somatic mutations to explain some of these unexplained inflammation. And thanks to my team, who has been working hard. You know, this is a sunny day in Leeds. We do get most, you know, occasional sunny days like this, and it was a good time to take a photo. So happy to take any questions.
Thank you, Dr. Savic. We are waiting for your questions live from microphone and via EULAR app and Q&A section.
Sanisha, while people are coming up, you're talking about pre-rheumatoid there. What about established rheumatoid and other diseases? What impact does inflammation have? And we know that chronic rheumatoid has LGL, so it's an adaptive immune or T cell or side of the equation.
No, absolutely. I mean, you know, we are hoping to start looking beyond, you know, to the whole rheumatoid timeline, because, I mean, the question is, I guess, you know, what comes first? Is there some sort of inflammatory trigger that maybe triggered some of these mutations, or is it that the mutations are the ones that are driving inflammation? So this concept is yet to be fully established, what causes what, and whether there is actually, or is it just random? But maybe there is enrichment, particularly of these mutations, particularly in patients who become resistant to multiple therapies, and which is something, as you know, we're very keen to look at.
Thank you for a wonderful talk. Just wanted your view on which mutations outside of the Vexus mutations you think will lead to diagnoses of new autoinflammatory diseases, and do you think multiple mutations, which we do see sometimes on next generation sequencing, can prognosticate in terms of worse disease?
No, that's great. Thank you for the question. So there's an interesting picture evolving, and some people have published this already, and we found this in our patients, particularly with this acquired NLRP3 mutations. You can get, for example, TET2 mutation, which does allow for the clonal survival. So it gives the clonal advantage survival. And co-mutation between TET2 and some of these other mutations allow for the clone enlargement. Other people have published, we have a patient who we had followed for a number of years, who started with a relatively treatable inflammatory illness, which we treated with, single dose of anakinra. But in 5 years from the original presentation, she progressed to now need, well, she's sadly passed away, but she then needed triple the dose of anakinra because the clone enlarged to the point where, so this sort of interplay is something we're just beginning to understand, really. Thank you for the question. Yeah, okay, thanks.
Thank you. We are going to the third speech, and our final speaker is Associate Professor Adam Handel from the Oxford University. He leads the autoimmune neurology research group and consultant neurologist in here. And we are starting to another interesting session and insights from AI and neuroimmunology.
Great. Well, thank you so much for inviting me, particularly as an interloper to this rheumatology meeting. These are my disclosures. So I'm going to talk a little bit about AI and its role both within neuroimmunology, which is the area I work in, but also many of these concepts, I think, can be extrapolated to other areas of medicine and basic science as well. And I'm going to split it down into a very brief introduction to AI, very topical at the moment. I do a lot of bioinformatic work, but I wouldn't claim to be a computer scientist, so I'm going to try and make it reasonably straightforward conceptually. I'm then going to show a few examples from our own work and the work of others in where AI has been usefully applied to basic neuroimmunology. I'm going to look a little bit at how that's also started to be used in clinical areas of neuroinflammation, and then I'm going to have a few thoughts at the end. So first of all, AI has actually been around for quite a while. You can see here a timeline of the different AI and machine learning associated tools and algorithms, and it goes right back to the early 1940s through to 1980s here. There are lots of different types of artificial intelligence. They're used for very many different tasks in many different ways. The capacity of these to perform tasks, as we're all aware from our use of AI in the day-to-day workplace and in our own lives, has massively expanded over time. And I think what some of the areas that has highlighted as issues, which we'll come on to later, particularly in relation to medicine, is what the AI can be trained on, because training is obviously absolutely key. If you can't train the system to do something, then it's not going to be functional. And what as well has changed over time is going from AIs that can recognize patterns within data and separate groups out into ones that can make new findings on novel data sets. And we're going to talk about some of the limitations of training and test data sets. But the way many of these studies work, just for those who are not aware, is that you'll have a large data set, and you'll train your machine learning algorithm on part of it, and you'll then keep part of it back so you can test the accuracy of the machine learning predictions on those. It's used in lots of different areas, and you can see healthcare here. It does lots of things from predicting disease, diagnosis, through to predicting outcomes of treatments. And there are some issues here, as you can immediately think, because an AI is mostly designed to give you an answer. It may not be the answer that you want, and so you need to make sure, as shown in this example from the New England Journal a little while ago, that actually it can express appropriate degrees of not knowing something before it can be used in medicine. There are a few different divisions that you can make amongst artificial intelligence approaches. So here it's sort of separated into quadrants, and you can either have input data into the machine learning where you annotate it. So that's where someone or something goes through and decides, is that cell type A or cell type B or disease A, disease B. And then you can train your machine learning algorithm to separate those two out, either by pre-curated data you gave it, so that might be abnormal MRI, yes or no, or you just feed it, as you can see on the deep learning thing, the MRI, and then it then extracts its own features out of there. But you can also give it non-annotated input data, and I'll show you an example of that later, where actually what the algorithm is doing there is trying to separate out meaning within the data that's not immediately apparent. And just to start off with a very general medical scenario, which is very relevant to all of us. So this is what happens when you give patients access to machine learning. How does it affect their decision-making parameters? And you can see the design here, that a load of doctors designed various vignettes with appropriate diagnoses and what they call disposition. So that's, should the patient go to their GP, do nothing, go to A&E immediately. And actually, when you give the AI some vignettes written in the same way as the training data, but not known to it, it's really quite good at predicting the diagnosis and also relatively good at telling the patient what they should do. However, when you then give patients the access to this AI algorithm, so here ChatGPT, for example, and give them their patient vignette and say, well, put your symptoms into the chatbot and then see what it says, actually the accuracy of its diagnosis and what it tells the patient to do is no better than if you didn't give the patient access to that. So this shows one key limitation with this is that you have to know how to use these tools or else they're worse than useless. Okay, so if we come on to the basic science bits first of all. So one of the areas that I'm particularly interested in is the thymus. And just to give you a little refresher on what this is, it's an area that selects developing T cells and they undergo waves of positive and negative selection. As you can see here in the cartoon, they go through the cortex and it selects for developing T cells that bind something, but then in the medulla, they get directed either into a Treg lineage or killed off if they're binding to self. And one of the reasons that's interesting to me as someone who studies neuroimmunology is that when you look in the single cell sequencing data set of these cells within the medulla that are selecting out thymocytes, many of those genes, as you would expect for a tissue-restricted disease, are present at low levels in low proportions of cells. So how you work out how the thymus is able to present those antigens to developing thymocytes is really important for neuroimmunological diseases. So we went on and we took those medullary thymic epithelial cells, which are the ones that present these autoantigens, and we looked at various chromatin modifications using ChIP-seq. So this is where you give them, you precipitate out regions of DNA and can work out what histone mark is around that area. And most of these tissue-restricted genes are targeted and promoted by something called AIR, which increases their expression levels within the thymus. And the question that we were really using machine learning for here, in this case you can see a neural network in the middle, was can we use those histone marks to predict whether a gene is or is not targeted by AIR? And you can see here on that we generate a neural network in the middle with a hidden layer in the middle, and that allows you to model not just the binary data coming in, but have interactions between different histone marks. And actually it achieves a pretty good accuracy over the null model, which is just basically guessing based on the proportion of genes. So that's one useful thing. But Mark already showed a lot of single cell data, and this is really where lots of the artificial intelligence tools are starting to take off. So this is just to show an example here of multiple sclerosis, which is one of the most commonly studied neuroimmunological, neuroinflammatory conditions. And in this, the authors took pieces of MS and control brain and used and captured the nuclei from those cells for sequencing, so you can get individual gene expression profiles for all the cells there. And they showed that, as you would expect, you can cluster them into lots and lots of different cell types, which is great. And when you look at the proportion of cells within each of those clusters, then you can see that certain cell types, for example, at the just around, around there, you can see that there's some excitatory neurons within layers 2 and 3 of the cortex. So those are present in controls, but not in MS, so presumably have been targeted by inflammatory processes. And you can also work out lots of different genes that are expressed along with the disease processes. But where AI starts to come in is the brain is a very complex organ, and so trying to go through and manually create all of those clusters is both time-consuming and also has the potential that you miss some variability that you just don't know how to look at. So if you bring in large-scale data sets, so here we're talking 33 million cells, and it will be increasing all the time, you can form these enormous cell type and organ-specific clustering. And the AI can very usefully be trained on that. So you can see here that you give it the various cell atlases where people have gone through and made their best guess annotations. It then learns how to interpret that from the gene expression profile of the cells. And you can then give it your new tissue where you don't know what the cell types are and see whether it can guess the tissues effectively. And actually, for some tissues, it works really quite well. So, for example, here you can see a confusion matrix for MS. So this is how well it guesses the human annotated clusters, which are down as gold standard. Now, you could argue, well, maybe the human wasn't great, but actually, I think you have to take something as a gold standard, so it's a very reasonable thing to do. And it does pretty well at bringing out the same cell types with both precision, so that's making sure that you call them accurately, and recall as well, so you don't call lots of false negatives. Okay, but you can go further than that, actually, because if you think about cell types, within that there'll be lots of heterogeneity of cell state, and knowing how to identify that can be quite tricky. So if you imagine here that you've got controls, they might be healthy, and then a case sample where they might be diseased. Well, in the disease sample, not all of those cells will be unhealthy. So some of them will be quite happy excitatory neurons, for example. And so machine learning is usefully able to, it makes a vector of diseaseness, as it were. So you can see that here. So this violin plot at the bottom here shows that control cells have this vector here. So that will be a vector composed of lots of different weightings from multiple different genes. Whereas in the case, you can see that there's a sort of a bump here. And the presumption is that these cells at the top are the ones that are, have something about them that's related to the disease process, whereas actually the ones at the bottom here are probably just bystander cells that haven't yet been affected by the process. So therefore, this machine learning algorithm is able to relabel these to draw attention to the more likely diseased cells. And that was used on the MS data set to show that, it's a slightly different one, it's in a mouse, but to show that there's a population of endothelial cells which, when you look at them without inferring this diseased versus non-diseased label, you can't pick up that there's any changes there. Whereas when you start to divide these into the ones that have been affected and not, then you can start to see that there's a clear transcriptomic and cell population effect in those cells. But you can go again beyond that, because cells do not exist in a vacuum. They're communicating with each other all the time. And we now have really rich data sets showing how cells send ligands, receive them on receptors, and then regulate intracellular pathways through transcriptomics. And you can essentially give a machine learning algorithm all of this data and pre-train it to recognize how you can see downstream effects of ligand receptor interactions. And when you're then looking at a comparison, that algorithm is able to infer connections between cells that are actually driving the process of interest. And we use that again in one of our thymus models. So you can see here, this is an RNA sequencing data set of thymic stromal cells, actually with the thymic epithelial cells removed for this project. And what we were interested in is this population of neural crest cells at the top and how those might signal to others to mediate age-related changes, because the thymus, as you'll all know, undergoes big age-related changes, even in a mouse, but particularly in a human. And by using the previous algorithm, we were able to see that the neural crest cells are very strongly signaling to endothelial cells and vascular smooth muscle to alter their transcriptomic profiles in a way that results in an aging process. We identified a few ligand receptor links here, and we were able to then show that there are actually reciprocal connections which drive the same aging process within the neural cell, crest cell population, because interestingly they migrate into the thymus, and then as far as we can tell, they transdifferentiate into other cell types and disappear. So that can really give you extra power. But thinking about immunology in particular, one of the things we really want to do when we're thinking about sort of the adaptive immune system beyond that is try and work out, well, for these T cells that we might get out in diseased tissue, what are they actually binding? Because that's a really hard question. And as you'll all know, they bind to an MHC molecule, which is presenting a peptide antigen of differing sizes, depending on if it's class 1 or 2. And there's a few applications of artificial intelligence to that have been used. And just as I showed you in the quadrants above, you can either have a supervised or unsupervised model. So in the supervised one, you essentially feed in a load of T-cell receptor sequences which are annotated by the peptide MHC molecule complex that they bind. That then generates a machine learning algorithm which you can then feed in with other unknown pairings in the hope that you can infer what antigen specificity there might be. Or you can just feed it the TCRs and then it looks for similarity between these. And that can actually be quite useful because if you're in a disease state, such as synovial fluid or cerebrospinal fluid in my case, then you would expect that large clusters which do not appear in healthy controls are likely to be antigen specific. But there are problems with particularly this supervised model. And when you look at the number of TCRs that are known to have antigenic targets, this is in thousands here, you can see that we're really good at knowing viral TCR antigen pairings, but we're absolutely rubbish at human peptide TCR pairings at the moment. That's clearly changing. And that's a real problem because if we're dominated by virus TCRs, we then can find it very hard to extrapolate that into the autoimmune field. And you can see that so this was a recent paper that looked at all the different AI methods of predicting TCR antigen MHC pairings. And they perform relatively well when you train them up on TCR epitope pairings that they know about and then introduce another one. So you can see here they get reasonable accuracies. This is sort of like an area under the curve for precision and recall. However, when you go to what's probably the more biologically interesting question and say, well, are there unseen epitopes, so ones that you haven't trained the model on that you can fish out? They're absolutely rubbish. They do just chance levels, really. And part of the reason for that was highlighted in this really nice paper a little while ago, which was looking at how many peptides would we have to know the TCR pairing of to be able to say that for a given peptide, when you were looking at an unknown one, you'd have a target known within one amino acid edit distance. And actually the numbers you can see here, these are all class 1 alleles, are absolutely enormous. So we're nowhere near the size that we need in terms of data sets yet. And that's really important because it gets even worse because when you look at these models again and say, well, okay, for an antigen TCR pair, how does it perform when we've got, say, over 300 TCRs that we know bind that antigen versus 10, you can see that it starts to dip down towards just above chance levels as we get fewer TCR pairs. So we're going to need many more than those millions that we saw before. So there were some earlier hopes. So this is a pan-PEP. So this was a TCR peptide MHC prediction. And the reason I put this in is it shows one of the perils of these papers when you think about how you train them up. And what you need to do for these models is you need to give them the positive TCR. So you've got a TCR and you've got its antigen of interest. However, what they've done in some models is that they say, well, we'll give it the training data and then we'll give it a background TCR data set that we assume is non-antigen specific, which is probably a fair assumption. But unfortunately, these models are very good at learning the differences between the positive TCRs and the background data set. So they don't actually learn anything about the antigen specificity. They just learn to distinguish the two data sets, which is not what you want. And when you take this quite good accuracy here and shuffle the background in this way, it falls basically to chance again, which is unfortunate. We've used some non-supervised clustering. So you can see here, this uses a sort of like an AlphaFold 2 prediction of TCR shape and clusters with a load of TCRs from patients with GAD, antibody-associated neurological problems. And so this shows one of the literature known GAD TCRs laid over one of the known, or a cluster of our CSF ones. We haven't expressed this yet, but obviously this is one way that you can prioritize TCR targets for functional work. So just to finish up on the basic side of it, so I think that the AI methods are really quite promising at understanding many aspects of single-cell biology that are not immediately apparent. but using them for further downstream antigen prediction is really challenging at the moment. And I've already mentioned many of these limitations. If you take that to trying to model antibody-antigen pairings, it gets even more difficult because you're not even constrained about where they can bind on the antigen. So if we just briefly then, for the last couple of minutes, talk about the clinical side of this. So in neuroimmunology, this has basically been applied to multiple sclerosis because you need many patients to do it, and so you need a common-ish disease. And it's largely been used either in classification, so do they have the disease or not, or prognosis. And most of the studies have looked at neuroimaging, but there are some coming through on biomarkers as well. This is the sort of thing that you might see. So you feed into the AI MRIs. So this is a flare, but they also fed it in the T1 as well. And then what you can read out of the machine learning is an attention map. So this is basically where the algorithm is looking to get its data on trying, in this case, to predict good versus bad outcomes. And then you can form these sort of maps of which bits are interesting. So here they were inferring that the frontal cortex and parts of the cerebellum were important in determining outcome, which makes a lot of sense. You can also, this is from a recent paper looking at about 1,000 patients, show that algorithms are pretty good at distinguishing MS from healthy controls, as are neuroradiologists, of course. It does reasonably well at predicting progressive sort of disease as well, or at least calling it from the MRIs, which is a bit more useful. When you then try and take it to predict the EDSS, so a disability scale, this shows some of the subtleties here. So some of the algorithms, so support vector machine learning, does okay. The random forest-based ones are much, much worse. So you have to be a little bit careful, and it's good that they use multiple ones. This is, again, looking at progression without inflammation from a sort of a prospective study looking at time 1 and time 2. And what you can see is that when you give it these clinical and paraclinical parameters, it's fairly good at deciding whether a patient will get progression or not. But what's really dominating it is the disability trajectory anyway, which is clinically apparent. So I think many of these will need to have multi-parameter data fit in to be more useful clinically. And there are studies doing that at the moment. So just to finish off, so this was a very useful paper looking at AI in neurology, and it suggested that we have a very clear progression of validation of these algorithms if they're to be used clinically. And I think it's really important, actually, that ongoing validation is done as well, because AI tools can potentially creep as the demographics of the population change, and so someone would need to keep an eye on that. I've told you a little bit about information leak from training data sets to test data sets. That will be clearly important. People have talked about black box interpretability. So that's the idea that it's just like a black box. It's making a decision. You don't know why. That might not actually be terribly a bad thing, because people have shown that gut feeling in GPs is a really good thing, and that often can't be boiled down to specific factors. But clearly we need a bit more oversight with AI. This is an acronym meaning garbage in, garbage out. So these processes are only as good as the training data. You feed them. And AI bias reinforcements is really important. So that can, because they trawl through a lot of our background information, they can reinforce societal biases, and we need to be careful of that. And finally, if we develop a model on a population, we need to be careful about taking it to another cohort. So you couldn't have an AI presentation without something generated by AI. So here we are. So I asked it to model me 3 different futures of human interaction with AI. Either we closely supervise it, we do minimal supervision, and we do virtually no supervision. And it's produced quite a nice graphic here saying that, you know, it's better for us to be involved, which is great. And then I said, well, you seem to be a bit kind to me. You're sort of pandering. So can you go a bit more extreme and project that into the future? So then I got this, which is looking less promising up here. AI is God, humans are obsolete. And then I said, well, one more, please. And then I got this. So by this point, humans are extinct now. Humans are optional in the middle option. So I guess we probably want to go for option number 1, I would think. So these are all my collaborators to acknowledge, and I always finish up by acknowledging my family who've put up with me for many years. So thanks very much for your attention.
Thank you for a nice talk. We are waiting for the question. While we are waiting, we have a question from the chat. Someone asked, how do you control for accuracy of your AI models if you don't have a gold standard?
Yeah, I mean, that's really difficult, isn't it? Because we're assuming that the humans are the gold standard often, or the existing diagnosis. So it's a real, you end up in this sort of circular argument where you don't really know exactly what the gold standard should be. I think it's a really good question. I hope it wasn't asked by an AI chatbot, but I suppose we'll never know, will we?
Please.
Hi, thank you for the talk, very nice talk. So I was wondering, I tried to follow the logic behind the case versus control single cell RNA-seq data that ends up with this binary system, disease versus not diseased. How do the transient phenotypes, cell phenotypes, fit into that might not really exactly match the one or the other condition?
So, you're talking about the one where we sort of binarize it into disease affected and not?
Yeah, yeah.
Yeah, so I mean, I suppose you have to be, you sort of have to be careful. The idea is that it's showing stuff that would normally cluster together, but then you're separating out the disease process ones specifically. But I always do slightly worry about these tools that you may be reinforcing what you're expecting to find, because you're delving right down into the information. And you're looking for a vector that will sort of fulfill your hypothesis, aren't you? You're saying there is a difference between there, it's just too subtle to see. So I suppose it's important then to go in when you've found that population and check that there are real, believable transcriptomic differences that would explain that.
All right, all good. So Adam, thank you very much, and thanks to Mark and Sanisha as well. Thank you all for coming, and from Ertugrul and myself, thank you. Good job.